This presentation is structured as we can see on this slide. First of all, I will talk about what are the copy number variants, with some anecdotes and information about them, talking about polymorphism and pathogenic events. Later, I will explain which is the validation done by us for the analysis of VarSeq software and his capacity to detect the copy number variants. After the validation, I will present a few interesting cases we resolved during this year and our experiences with them. Finally, I will show you my conclusions that I have found working on this project and which are my recommendations in the study of copy number variants. But firstly, I would like to introduce the laboratory where I'm working now, Reference Laboratory. Reference Laboratory is a Spanish laboratory that collaborates with more than 1,000 hospitals and laboratories worldwide, having more than 40 years experience in all kinds of analysis. In Reference Laboratory genetics, all types of studies are carried out: hereditary genetic diseases, studies of solid tumors and liquid biopsies, oncohematology, pharmacogenetics, etc. As we can see on this slide, we work with a large number of technologies, customizing the studies to the requirements of our clients, using for the current classification of the variants the international guides. Talking about copy number variants, nobody you can ask us to do CNVs in any panel of genes you need that. Once introduced where I work, I have prepared an introduction with some interesting data about copy number variants, as well as the evolution of this analysis during the last decade. First of all, what are the copy number variants? Maybe it is the first question that will come up to our mind, and I will not be able to define easily, with a description that we are all in agreement. As we will see, there are discrepancies in the literature. It's a type of structural variant which affects the number of genomic copies of a region. In some publications and definitions, we will see that they indicate that they are greater than 1 kilobyte size, but with the latest technologies in copy number variants analysis, this value maybe should be modified. These variants play a very important role in population variability, and we find it in most of the complex organisms. In many publications, it has been postulated its importance with the speciation factors as well as in the evolution. All this has led to the increasing related investigation of copy number variants in multiple research areas. In this figure, we can see different examples of copy number variants we would like to talk, as duplications and deletions. In this figure, we see a typical gene with 4 exons. In this case, we lose exon and having a deletion. And in this other, we have a duplication of exon B. Focusing more on the molecular level, I want to emphasize how copy number variant analysis have been introduced into the clinical diagnosis routine in the last 15 years. This allows us to increase our knowledge and understanding the importance of these variants in our genome. The more our knowledge increases in this area, the more we can learn about it. Having observed that in proportion, the number of polymorphic copy number variants in the genome is greater than that of polymorphic single nucleotide variants. There is a discussion in a scientific literature about how much they are contributing to this polymorphism. There are publications affirming that approximately 10% of our genome is formed by copy number variants, and when we compare genomes, we can see that can be more than 1.2% of difference between samples that can be detected due to these CNVs. With all this information, we can already predict that there are multiple copy number variants with frequency greater than 1% in the population. The most surprising is the fact that currently there are already about 100 genes that have been found with an homozygous deletion without any apparent adverse phenotype. Despite all this data, we must not forget that copy number variants are also a common source of multiple pathologies. I want to emphasize that we still don't know the effect of many copy number variants. Over the years and the increasing of the knowledge of copy number variants, we will be able to understand better this type of genetic variants and how they affect a person's daily life. I want to talk about some of well-known and well-worked examples at copy number variant level. These are DiGeorge syndrome. It's a good example of a well-known for a long time copy number variant. It was generated by a deletion that affects a segment of chromosome 22. Decreasing the resolution and talking about exonic copy number variants, we find an example in Duchenne dystrophy, where we see that between 65 and 85% of the affected ones by this pathology are related to deletions in the DMD gene. A strange effect in the phenotype related with a copy number variant event is related with the PMP22 gene, where, depending on if it presents a duplication or a deletion, we will find different clinical phenotypes, being Charcot-Marie-Tooth type 1 or hereditary process-sensitive neuropathy, respectively. Talking about multigenic syndrome, in development delays with a known etiology have been found that copy number variants where the cause is 17% of patients. As an example of the pathology of colon debris, where 95% of the cases are explained by a deletion in CANS-L1 gene. For the analysis of copy number variants, several technologies have been used over the years. One of the most classical is the karyotyping, but we will have to talk about very large events to be able to detect them with this technology. Currently, a widely used technique is the RAI-CGH, which allows us to analyze the whole exome, but its ability to discern changes is dependent on the number of proofs per region. It is needed a minimum of proof for its acceptance. Current designs have multiple proofs scattered through the genome. At a counterpoint, there are not many proofs in each region, allowing us only to see large regions affected. Exonic arrays have begun to emerge. They are a new kind of array that have 3 proofs by exome, but not having the capacity of analyzing many genes and with a high cost. In order to see copy number variants affecting specific exons of a gene, we must use the MLPA, which allows us a sensitivity related to an exonic level. But the greatest counterpoint we have with this technology is that you need to know which regions we would like to analyze, since the querying kits are for a single gene or a few additional regions. To try to solve the counterpoints of these technologies, in recent years, a new approach has emerged to analyze these changes in the hands of the next generation sequencing data, which was implemented thanks to the reduction in the cost of sequencing and the increased use of this technology. This approach allows us, with a single run, the analysis of copy number variants and single nucleotide variants, which can decrease the time to obtain a result. Depending on which kit used to make the sequencing, we can be able to analyze complete genomes. The great difficulty to this approach is the greatest computational complexity behind this analysis, requiring specialized tools for the correct detection. Currently, we are speaking of a technique that has a high sensitivity, but with a low specificity, generating a high number of false positives. We'll talk of this later and how it could be solved. There are 2 approaches based on the NGS data, one based on the depth of coverage, and other known as paired end mapping. This last is based on finding the discrepancies between the distance of the sequencing pairs. We were looking for a software that allows us the analysis of single nucleotide variants and copy number variants, giving us, with just a pair of clicks, clinical annotations and filtering of the data. It needs to have specific features. We wanted that the CNVs analysis were based on the method of the depth of coverage approach, which is based on a statistical normalization of the coverage depth. The choice of VarSeq was made because it offers us the features as a good quality price, and after successful validation, which I will comment later, we decided to work with them. One of the greatest strengths of this software is its malleability and moldability it has to our disposal, allowing the analysts to generate their own analytical workflow, being able to adapt the data show to the needs of each analysis, and performing customized and high specialized analysis. With the work created by our team, we verified that we can detect changes of at least a single exon with great sensitivity. In this slide, we can see an image of the data pre-analyzed by the software, observing the great capacity that it gives us due to the high number of options and calculations to our disposal. In these slides about what are the CNVs and how are analyzed, I would like to talk about our validation of VarSeq, showing you what are our criteria about the software. On these slides, I will present what are the idea at a level of sample selection, as well as which regions we have been used. Our validation of the software has been made from about 20 to 30 samples with a previously known copy number variant, either by MLPA or array. We carry out a blind analysis only knowing the sequencing panel previously performed, similar to what could be obtained from a clinical history. We have used samples with different types of events, even checking some anomalies. New batches of samples were introduced regularly in our analysis with a known event in order to better estimate the sensitivity. As previously stated, one of the first things that are detected in the analysis of copy number variants with NGS data is the high rate of false positive, so it is very important to work with a large database of controls. We currently have more than 2,000 loaded controls, achieving a higher and better normalization of the data. It is also very important the approach with the clinical data and the HPOs, the Human Phenotype Ontology, which help us to focus on the regions where we expect to find those possible CNVs. As I explained at the beginning of this exposition, many polymorphic copy number variants are found in the genome, so it is important to know the kind of affection we are searching. The samples that we decide to include for our validation followed one or multiple parameters that we wanted to verify. An idea of the parameters used was working with different sizes of changes, as well as different kind of events, such as duplication and heterozygous deletions. We also want to check genes with a high number of known CNVs, such as DMD, BRCA1, BRCA2, F8, etc., since they will be one of the common analyses that we will have to carry out. In addition, to force the analysis, we wanted to incorporate and analyze areas with a lot of variability, such as the polymorphic region of the CF-ACR1 gene and CF-ACR3 gene, or a PMP22 gene, which, as explained before, is pathological, but with a relative high proportion in population. Multiple samples with this kind of events were being incorporated into the controls. As we can see in the figure below, the analysis of these regions may seem complicated, but thanks to a high number of controls, the software is able to design and identify those CNVs on these regions. In this table, I show some of the regions and alterations used for this validation. As you can see, there are all kinds of events, including different size and location. Examples of the genes are F8, BRCA2, MSH2, F8, another time, are having events like duplications, homozygous deletions, and heterozygous deletion of one gene, of one exon, or complete genes. As I have been emphasizing on many occasions, it is very important to use a large database of controls, included in the same run, as well as data of multiple runs, greatly improving the analysis of the regions involved. In the example below, we see the same region, but on the analysis on the left with few controls. In this, the event is not well detected or may have detection problems, giving us values that could lead to an error that could give us a false positive. But with more controls, the false positive disappear, as we can see on the figure on the right. An important point is that during 2018, all the detected changes, whenever possible, have been checked by MLPA or any other technique that will allow it. To finish this swap validation, I have received different software inputs. The values that are decided and used for the acceptance or not of an event are variable depending on many factors, such as the average coverage, number of controls, region, sequencing kits, etc. So each center with no experience must decide and choose the cutoff points. And that's how we have done this validation of the software. I would like to comment some of our cases, explaining our experience as well as the usefulness and the progress achieved with incorporation of CNVs in our analysis. During the last year, 275 CNVs analyses were performed on samples in which single nucleotide variants were previously analyzed and has not been reached a conclusive result. In 12 of the samples, we were able to reach diagnostic conclusions thanks to the analysis of the copy number variants, increasing the number of cases solved by 4%. In the following table, we can see that the events detected prove that our clinical sensitivity reaches up to a single exon, finding events in a large number of different genes associated with multiple pathologies. I would also like to comment that at the beginning of this year, 2019, 4 new variants have already been detected that could explain the patient clinic diagnosis. 2 of them are important to complete the study of validation, confirming the correct detection of copy number variants, since they are duplication, events that we have not found in our clinical samples in 2018, although we did in our validation controls. One of them is a complete duplication of the BRCA1 gene, which we are studying to verify if the size is higher than only the gene, since in the used sequencing kit, there were not adjoining genes to confirm the true size of the event. The second duplication will be very interesting since it affects only a single exon. Now I can confirm its veracity, or if it's a false positive, since at this moment it is being corroborated by MLPA, although everything points to its truthfulness. The last relevant variant that I wanted to comment is a deletion in TNXB gene, which have a great homology with the TNXA gene. This case will be discussed later with more detail. To finish this data introduction, I would like to advance one of the possible areas in which we will start working, once we collect all the necessary information and validate it. Pharmacogenetics associated with duplication and deletions in some CYP genes, such as the CYP2D6 gene, where many CNVs have been described affecting the metabolization. People with a duplication affecting this gene will be quick metabolizers. Now, I will explain with more detail some of the cases. Firstly, I will talk about a patient with suspicion of severe bilateral prelocutive sensorineuronal hearing loss. In addition, he was a patient with consanguinity. A panel of inherited recessive hearing loss of 39 genes was performed, but it was inconclusive. A month later, it was decided to perform a copy number variant analysis where a homozygous variant was detected in exon 2 of the RDX gene that will confirm the diagnostic hypothesis. As there was no commercial kit to prove it, a Sanger versus control was carried out to confirm that it wasn't a sequencing error in our sample. Then a segregation analysis was recommended in the parents, and it was found that both were carriers of the variant detected. At the clinical level, this variant correlates with the affection of the patient, since we can see that this gene is related to an autosomal recessive deafness type 24. I would also like to point how in 2018 another group detected the same event in one sample of an analysis of a Spanish population. As we have seen in these two slides, the deletion in the index patient is corroborated and the carrier status in the parents was very clear. For future pregnancies will be very important prenatal analysis, since there is a 25% possibilities that future children are affected by this disease. Another interesting case is found in a patient with chronic hypomagnesemia diagnostic in childhood with secondary hypocalcemia. It does not refer other cases in the family. We made a panel of hypomagnesemia with secondary hypocalcemia centered on the TRPM6 gene. The analysis was not conclusive. Subsequently, we were asked to analyze the copy number variants, where two deletions are detected in heterozygous in the TRPM6 gene that could confirm the diagnostic hypothesis depending on the segregation. In this case, the realization of segregation was very important to check whether the variants are in cis or in trans. Additionally, in this case, we do not have a way to prove the CNVs. The data from the parents could help us to confirm its existences. In addition, I would like to highlight that in our database, we have never detected an alteration in these exons, which could let us to believe that it was real and not a specificity error. When the segregation is carried out in the parents, it is verified that both are carriers of the variant, confirming that in the index patient, the variants are in trans. In addition, it should be noted that TRPM6 gene is associated with hypomagnesemia with secondary hypocalcemia with an autosomal recessive inheritance, which could confirm the diagnostic hypothesis which it was worked. This particular deletion are not in any database, but we found multiple data of deletions causing the pathology in TRPM6 gene. As we can see on these slides, each parent was a carrier of one of the variants, confirming the diagnosis. And as in the previous case, it is recommended that the parents, if they wanted to have new offspring, perform a prenatal analysis, since 25% of their children will be affected. The following case was advanced it previously. We have a patient with joint hypermobility of the spine, of which a possible Ehlers-Danlos syndrome is suspected, which, as in the previous case, the single nucleotide variants analysis was not conclusive. When performing the analysis of copy number variants, a deletion in homozygosis was detected in the exon 35 of TNXB gene that will confirm the diagnostic hypothesis. This gene, as we can see in the image below, has a lot of background noise. This is due to the fact that many exons of the TNXB gene have a great homology with the TNXA gene. If it is not well analyzed, it is possible to don't see it or think it's an error. As shown in this slide, it is categorized as heterozygous deletion, being in reality a homozygous deletion. The TNXB gene could match the patient clinical condition since it is associated with the classical Ehlers-Danlos form. Doing the proof by MLPA, it was possible to verify that the exon that apparently was deleted was confirmed. Although, as in our NGS data, the exon adjacent to it, which have greater homology, could not be analyzed. This shows a weak point of all these techniques, the pseudogenes. But I wanted to show to you, since despite all the background noise generated by these homologous regions, non-homologous regions can be analyzed perfectly without being negatively affected by them. We are currently working on a dilution of this sample to be able to validate the detection of this kind of variant of a single exon between homologous regions even in heterozygosis. The last case I want to tell you about is a patient with possible Marfan syndrome. So a Marfan syndrome panel of 10 genes is performed without obtaining a conclusive result in the analysis. After 1 year, it was decided to carry out the analysis of copy number variants where heterozygous deletion is detected in exon 2 of the FBN1 gene. That will confirm the diagnostic hypothesis. In addition, it was detected that this deletion also affects the CYP152 gene. As we can see on this slide, the overall corroborates the variant observed by NGS data. When looking at the reference for this gene, we see how this gene is associated with Marfan syndrome, and we found multiple references to the implication of genes adjacent to FBN1. This is a curious case of how multiple genes may be affected and how it could modulate or affect the clinical symptoms of this patient. This case could not have been completely resolved by MLPA because it could not discern the implication of the second gene. Finally, I want to tell you about the diagnostic difficulty for duplications. One example that we had is an asymptomatic 46-year-old patient with multiple family antecedents of breast cancer. A breast, ovarian, and colon cancer panel of 29 genes was performed, without detecting any variant that could be the cause of the pathology. When performing the analysis of copy number variants, we found that the BRCA1 gene was duplicated entirely. In the bibliography, we have found reference of duplication as a risk factor for breast cancer, but not having the capacity to know where the duplication is located, we have to report as a pause. But why? It is because the great problem with the duplication is, where is the duplication? There are different possibilities. One of them is the duplication of the entire gene could generate an extra doses of protein, giving us a gain of function. Maybe those duplications are inside the regulatory region or an intern of a gene, altering the function of this gene. In this figure, we can see two genes. In this first example, we see a duplication of the exon B inside the gene, altering the functionality of the gene. In this second example, the exon is duplicated in an area that does not affect any functionality of any gene. And in the last example, the duplication is inside another gene, and this will affect the functionality of this gene. To conclude my presentation, I would like to tell you my conclusions about all the data showing the importance of copy number variants in the clinical diagnosis. And as we have seen, how many of these cases have a common thing. Many of them have a long time to reach a final diagnosis. During 2018, in 4% of the samples with a conclusive diagnosis in the analysis of single nucleotide variants, the analysis of copy number variants was performed, the cause was detected by them, and increasing the diagnostic efficiency of 0.3% over the overall samples analyzed for us in 2018. We estimate that if in all the cases without diagnostic conclusion by a single nucleotide variants, analysis of copy number variants was performed, an increase of 3% in diagnostic efficiency would have been obtained. A 3% may not seem a lot, but it's important to not forget that we are talking about patients that will receive a final diagnosis. The results obtained confirm that the inclusion of the detection of copy number variants by NGS data in the routine of genetic diagnosis allowed to increase the diagnostic efficiency of FARET. In the experimental examples, we have seen that not performing the analysis of single nucleotide variants and copy number variants in parallel increased the time of the diagnosis conclusion in about 1 to 3 months, having cases with greater than 1 year to obtain the diagnosis. Therefore, with our experience and for all that was commented in all these slides, our final conclusion is not only the recommendation on performing the copy number variants in the non-conclusive analysis of single nucleotide variants, allowing a greater diagnostic efficiency. It's performing the analysis of single nucleotide variants and copy number variants in parallel. Since in many of the cases showed, they get to lengthen months or years to obtain the final diagnosis. With the analysis in parallel, an increased diagnosis efficiency is obtained in less time. Here, I have left part of the bibliography used in the presentation. Thanks for your attention, and don't hesitate to ask me any question you have. As you can also send me these questions to my mail, and as soon as I can, I will send the answers. And as